Good morning, everyone. Hello, Rahul Pandey. After yesterday's CP, you are the first one here. Honey, why you miss CP? Tejasvi also. Okay, guys. Today our lecture is about coronavirus, but now not about the immune response or vaccines against coronavirus. So you've heard already the lecture about the immunological aspects of COVID infection. Today's lecture is more like virology. So it covers a bit of virological routine, the description of the virus and how this virus replicate, and also pathogenesis of the virus. But you have to understand, for this disease that is called COVID-19, we cannot really separate the pathogenesis of the disease and the immune response, because the tissue damage is mainly to immunopathogenesis. So, and because we are working without the break, so 1:30 sharp we have to finish, because you know I have to run to the next class, that is CP. So that's why we work without the break, okay? So, and at the very first slide, you see again the picture of the under the scanning electron microscope, where we see the very nice halo around the virion that is red here. This false colored scanning microscopy. And this is the virus of middle size. So the diameter ranges from 60 to 140 nanometers, but then the outer surface studded with the distinctive 9 to 12 nanometers long spikes that have the shape. They give the virion the appearance of the solar corona or solar crown, not king's crown, but solar crown. So this appearance under the microscope gives rise to the name of this viruses group. Okay, so I would like to tell you a few words about the history of the human coronaviruses. It had not been started in 2019, not at all. So the first coronaviruses had been found in the mid-60s of the previous century, 1960s, right? And the first two human coronaviruses that were called HCoV were identified. Those names mean not so much for you. Just two coronaviruses that can cause infections in humans. They had been studied extensively to the mid of the 80s of the previous century, 1980s. So what they did, by infecting the healthy volunteers, the researchers learned that infection with these two human coronaviruses result in a very mild diseases or no diseases at all. If disease was symptomatic, so it used to cause a common cold and maybe slight GIT problems like diarrhea, but not so much else. So, and since that mid of 80s of the previous century, human coronaviruses had been considered to be relatively harmless. Can you imagine? So, and they were considered as very mild respiratory GIT pathogens, so we didn't really pay that much attention on them at all. And this image was roughly disturbed when severe acute respiratory syndrome, that was called SARS, coronavirus was introduced into the human population in the winter of 2002 and 2003, and again it had been started in China. SARS-CoV that time causes a severe respiratory illness, unusual, atypical pneumonia with high morbidity and mortality. Morbidity, this is the incidence of the disease, and mortality, this is the incidence of the deaths. So, and my dears, this I will show you a couple of slides that I did in 2003 for students' lecture. So, because that was very new that time. So, SARS was recognized as one of the latest in a series of new human infectious diseases. It was recognized as a zoonotic, transmitted from animals to humans, deadly form of atypical unusual pneumonia. So it emerged in southern China in late 2002, but then it rapidly spread to different areas of the Far East and several other countries around the globe. So it spread pandemically. So the global air travel has facilitated the international spread of SARS. And SARS caused restrictions on international travel and economic downturn in affected countries. And that time, it was really something really very new. And the WHO officially declared on 6th of April 2003 that SARS-associated coronavirus never before seen in human population, and it is recognized as a cause of SARS. So, what was the hypothesis of the origin of the virus? It appears that the SARS virus, number one, as we know now, showed the 99% of nucleotide sequences similarity to the surface pass protein of the human SARS coronavirus had been isolated in Guangdong, China, from masked palm civet. Can you see this very nice, very cute animal? Paguma larvata. This is the masked palm civet, cat-like mammal closely related to the mongoose. This civet cat is blamed as a source of SARS, and this civet cat is regarded as a delicacy in China, so they eat it. And it is believed that humans become infected as they raised and slaughtered these types of animals. That was about SARS-CoV. And that again, this is the picture taken from the newspaper on the 2003, because that was so-called emerging viral infection. So that emerged from nowhere in the population and human population, never seen it before, and it has been introduced and it also caused fear. Can you see a fear in your eyes? And that was the respirators, also the masks that they wear. So, and then that was the first human coronavirus that caused pandemic. So later on, one more human coronavirus appears that initially had been called Saudi SARS. It was at the beginning of the researching of this virus. So it was considered very similar to SARS. So it was then later on it had been called MERS coronavirus, not because of the Mercedes, of course, my dears. So MERS, So because of Middle East respiratory syndrome, coronavirus. That's supposed to be a new member of coronavirus that causes infections in Saudi Arabia. 80% of all infections had been caused there. Then Jordan, Egypt, and some other countries. It appears to be distinct from SARS-CoV and distinct from other human coronaviruses that could cause common cold and some other mild disease. And until the 23rd of May 2013, and it appeared in 2012, MERS coronavirus had frequently been referred to the SARS-like Saudi virus, but since 2012, we know now that around 27 countries have reported cases of MERS, Middle East Respiratory Syndrome. It is a zoonotic virus transmitted between animals and people. And studies have shown that humans are infected through direct contact or indirect contact with infected dromedary camels. So you can see a member of, you can see this animal here. So that's supposed to be the vector for transmission. So the MERS coronavirus in humans showed the strong tropism for unusual cells, non-ciliated bronchial epithelial cells. And it has been shown to effectively evade the immune response and antagonize interferon that the very good cytokine produced within the human immune system activation, again with very strong antiviral activity. So this tropism is unique in that most respiratory viruses because they usually target ciliated cells. And due to the clinical similarity between MERS and SARS, it was proposed that they may use the same cellular receptors. So the exopeptidases, angiotensin converting enzyme 2, and however, it was later discovered dipeptidyl peptidase is also known as CD26 as a functional receptor for MERS-CoV. So there were two killers before. SARS coronavirus infected 8,000 people and killing about 10% of infected, and MERS. But then this one disappeared from the population at 2003 and never seen in human population since that time. So it was contained within 7 months and a half. MERS has never stopped, so even now we have a very low but permanent incidence of the disease. And since arising in 2012, it infects 2,000 people and a half roughly and kills 35 of them, so percent of them. So the lethality of this virus is much higher. And again, a novel SARS coronavirus appeared, and now it infects more than 65 million people, if I'm not mistaken, a bit more already, and kills more than 1.5 million of them. And my dears, it is not going to stop. So, and here you can see a summarized table of human coronaviruses and their origin. And you can see here that mostly we have a natural host that is somewhere in between the human and where the coronavirus may occur naturally. So this is natural reservoir. What animals may serve as natural reservoirs? Those animals in which the disease is not really symptomatic, that may harbor viruses without, or some other microbes, in our case coronaviruses, without really having any damage to the population of these animals. So, and here you can see that some viruses are designated with yellow arrows, and some viruses are designated with the red arrows. So these are the viruses that indicated as yellow, with yellow arrows, are those that cause very mild disease in humans, and those with red arrows, those that cause quite severe diseases in humans. And all new coronaviruses, SARS-CoV, MERS-CoV, and SARS-CoV-2, they are labeled as red. And you can see here that the most popular natural host, and of course this is just hypothesis. So it's not really proven yet completely. But because the researchers isolate and perform the nucleic acid um, analysis of all strains isolated from animals, they compare the similarity of those strains so they could develop the probable way of the origin of these viruses in the human population. I won't talk so much about the viruses that appeared before, and they are not very dangerous to human population. So, taking into consideration that two coronaviruses appeared in 2004 and 2005, but they didn't cause any really uh, importance in the social economical state of the world because they were really very mild. But those three that appeared since 2003, SARS, MERS, and SARS-CoV-2, so they all have most probably the natural hosts, the bats as natural hosts. And then they get to human population through different types of animals. So you mentioned already civet cat for SARS-CoV as intermediate host, and camels as intermediate hosts for MERS. But then it would be correct to put a question mark here because you can see here these are pangolins. They really developed the disease caused by coronavirus. So, and then we don't really know. That is just one of the versions. What kind of animal? Maybe most probably pangolins are the intermediate hosts for the bad coronavirus that give rise to the SARS-CoV-2. My dears, this is not proven yet, but then this is a table taken from the very high-ranked scientific journal Nature. So then, most probably, we can rely on it. So, originally, the virus that we discussed today, mainly SARS-CoV-2 coronavirus, was called 2009 N coronavirus, novel or new novel. But later, the International Committee on Taxonomy of Viruses on February 11, 2020, officially named it as SARS-CoV-2 because of its genetic similarity with SARS-CoV-1. SARS-CoV-2 causes respiratory illness, and WHO named the disease as COVID-19. It is highly contagious, primarily transmitted among people through respiratory droplets and contact routes. So WHO declared the COVID outbreak a public health emergency of international concern, and later on March 11, it was recognized as pandemic. Few words about the classification of coronaviruses. They belong to order Nidovirales, and the family Coronaviridae with the similar appearance under the scanning microscope with the halo and solar crown. And in this family, there is a subfamily coronavirus, and based on serology and phylogenic relatedness, so they are subdivided into four genera from alpha to delta coronavirus, alpha, beta, gamma, delta. We are not interested at all right now. Maybe in future, I don't, you never know. But right now, gamma and delta coronavirus don't give rise to the human infections. And all viruses that can cause infections in humans are uh, mm, assigned to the alpha coronavirus, alpha, and beta coronaviruses. And especially beta coronavirus genus is responsible for giving us so new emerging coronaviruses, SARS-CoV, MERS-CoV and SARS-CoV-2. So they are all beta coronaviruses. Now let's take a look, and I think I demonstrated this picture already. So I just want to remind you that this is the Baltimore classification. Mainly is based on the structure of the viral genome and strategy of viral genome transcription and translation. It means copying and expression in the human cell. And type 4, this is the group of viruses that have single-stranded, positive sense RNA genome. So, and what you can see here. So we go ahead with the structural characteristics of this virus. And this is a very nice picture that shows very important viral proteins and also the viral genome. But that we'll see later on also. So, a positive sense single-stranded RNA, and we may call it messenger RNA because the coronavirus RNA can be can undergo direct translation in the human cell ribosomes without any modification. So that's why we call it messenger RNA. And the genome of coronavirus is the largest among RNA viruses. It is very long, around 30 kilobases, and it encodes 13 open reading frames for translation of the proteins, and this is abnormally high rate over the open reading frames. Let's take a look at the structure of the virion. You know that this is the enveloped virus. Let's take a look at the envelope. This is the lipid membrane taken from the human, mainly endoplasmic reticulum. And then several types of proteins are inserted into this membrane. The most important one is a spike protein. This is a glycosylated protein made up of two monomers, S1 and S2. And this is trimeric protein, binds usually to angiotensin converting enzyme 2 on the host cell, and it needs the proteolytic activation of one more protein, transmembrane protein receptor serine 2 protease. So, and that is very important because this is the binding. So on the spike protein, we have a binding site for binding with the host cell receptor. And usually it is it is not so small, up to 10 nanometers in size, and around 100 trimers are found on one virion. So then the second type is membrane protein. it also incorporated into the membrane. And the very important function of it is interferon antagonist. It inhibits the activity of first type interferon. So that's one of the ways to escape immune response. And we have around 2000 copies per virion. So the third one, you can see the very small one, envelope protein, we designated as E protein. So, and this is also called viroporin, host that may cause the host cell lysis, and we may have around 20 copies per virion. And so what is unusual for this type of the viruses, we don't see by now any membrane protein, my dears. Maybe within a year the insight on the structure will be changed, but by now we don't think the membrane protein is, oh sorry, the matrix protein is there, so we don't see it yet. So, but then nucleocapsid is of helical symmetry. Can you see the helical symmetry? When the N protein, nucleocapsid protein, binds tightly with the nucleic acid to form ribonucleocapsid. And the N protein is also interferon antagonist. And we may see around 1000 copies of this protein per virion. Now we take a look at some more structures or some more functions of these proteins. So again, the S protein, this is the one that binds with host receptor. And S1, I said that it is divided into S1 and S2. These are vital functions to regulate the virus-host interaction. And S2 mediate the membrane fusion and viral entry. So we have the particular functions of the S1, S2. So membrane protein, this is not matrix, membrane protein is involved in viral integration into the integration means the fusion with the endosome or fusion with the cell membrane. E protein facilitates assembly, envelope formation, and budding. So this very small E protein. And N protein is critical in encapsulation of the genome, packaging the nucleic acid into the virion, and effective viral replication. So these are the major viral proteins. Then one more look at the S protein, and you can see here this is a configuration of the spike. So, and here we have a so-called the site or domain that is called receptor binding domain, and this is configuration of this receptor binding domain that happens on the edge of the spike protein. So, and then this is angiotensin converting enzyme 2 receptor that match with the receptor binding domain. So that what kind of interaction make or responsible for narrow tissue tropism of this virus. So, but then with some studies, it is shown that SARS-CoV-2 might exploit other host factors such as vimentin, a cytoskeleton protein, and lectins, a sort of glycoproteins to mediate viral entry. Okay, what did I say? I said that AC2 is the principal receptor for SARS-CoV-2, but then a part of this receptor, it may, we believe now that it may enter through a cytoskeleton protein vimentin and lectins as glycoproteins into these cells. So some alternative. Few words more about the genome. So you know single-stranded RNA, messenger RNA, but then coronavirus genome have the longest RNA virus genome known, and it encodes 4 structural proteins. Can you take a look here? This is S protein, this one, then M protein, this one, E protein, this one, blue, and N protein, that is inside 4 structural proteins. Apart from this, it encodes information about 15 to 16 non-structural proteins. Can you imagine? That's so many. So, now let's talk about the viral replication, and we will underline the unusual stages or unusual events in the viral replication also. So we underline this. So we mentioned already that efficient host cell entry depends on the cleavage of, see, this is a spike, of cleavage the spike into C1 and C2. And this is done by the so-called transmembrane protein receptor serine protease 2, and that shortly called as TMPRSS2. And second, the fusion may occur or the endosomal cleavage may occur with the help of cathepsin L, which mediates virus cell membrane fusion at the cell surface or in endosomal fusion. So what does it mean? It means that by now we think that it may enter by 2 routes: direct fusion with the cell membrane, or it can be uptaken through the receptor-mediated endocytosis as a whole virus into the endosome, and then endosomal membrane lysis will occur, and anyhow, by the, if virus enters by this route or by the alternative route of endocytosis, so the viral RNA will be released, the viral genome will be released into the cytosol, right? So this is the result of binding, entry, and uncoating in the same time, because there is no M protein, so matrix protein. So even as a result of fusion, the RNA with the nucleocapsid protein directly will be released into the cytosol. What next? So once the RNA is released, it will be translated onto the ribosome directly, but then not the whole RNA will be translated, but the very small part from the first of the outer reading open reading frame, and the couple of proteins will be produced. So that's supposed to be cleaved by viral encoded protease. This is proteolysis, and you can see that those peptides are cleaved into the several proteins. So non-structural proteins, because they are not incorporated into the virion, and they just produced as soon as the M viral genome is released into the cell. So, and these proteins form a so-called replicase complex. So that is needed to start the genome replication. And several types of proteins are participating in this complex formation: a catalytic subunit NSP12 and 2 accessory subunits of NSP7 and non-structural protein 8. Which increase the RNA-dependent RNA polymerase template binding and processivity. So it means these proteins, including RNA-dependent RNA polymerase. But this enzyme is the major enzyme to perform transcription. So replication, and you can see the replicase transcriptase complex is formed, then replication begins. But then for replication, the special double membrane vesicles are formed based on the Golgi apparatus. So, and the researchers think that the development of these double membrane vesicles will help to escape the activity of interferons to protect the process of virus replication and their later on assembly from the activity of the interferons. So we go ahead. So the incoming positive strand genome then solves. So this is the same positive strand genome that had been used for the open reading frame mRNA production, open reading frame, translation. The same positive strand genome serves as a template for full length. This is the negative sense, and this is of different color, right? The pink. So for the transcription of full-length negative strand RNA. And this is subgenomic RNA. So it is called subgenomic RNA. So, and then subgenomic RNA will be sent, transcribed into positive sense mRNA, and then will be sent for the proteins translation. You can see here viral proteins translation. So structural proteins and then a lot of non-structural proteins and also will be translated. So this later on the viral maturation and assembly occurs again in Golgi apparatus intermediate compartments. You can see it here, right? Endoplasmic reticulum, Golgi intermediate complexes or compartment, right? And the surface glycoproteins will be expressed on the surface of these membranes. And finally, subsequent transcribed positive sense genomic RNA will be taken to the nucleocapsids and incorporated into the nucleocapsid, so the viral assembly occurs. And then viral maturation and release by receptor-mediated exocytosis will be released, or maybe by cell lysis in some cases. That are the most general stages of the viral replication. While we discuss the viral replication, it will be good to take a look at the same time to the targets in the replicative cycle that can be used as targets for the anti-COVID therapy. So, and my dears, because of the so high socio-economical and health input of the virus, so then a lot of studies are done right now, really a lot. If you take a look at the internet, there's so many candidates for treatment because a lot of research groups start doing it immediately, all together or separately. So that's why you have to understand that not all approaches are equally available from one side, and not all approaches are equally effective in the treatment of the COVID infections, because all those candidate of drugs are intensively used mainly in the animal models, okay? But then we just take a rough look at the approaches used here. First, to the drugs may target the so-called structural targets. You know the importance of the E protein of the coronavirus, and this is furin-like cleavage site of the E protein. If it is protected, then the cleavage will not occur. Then we have an intensive study, of course, to inhibit the angiotensin converting enzyme receptors to prevent the viral entry. And of course, this is a very good way to prevent the disease at the very beginning. And also because it needs the core receptor. So also some transmembrane serine protease inhibitors are under study to prevent viral activation. So then some drugs, for example, well-known chloroquine, that is the anti-malaria drug initially used as anti-malaria drug. It is believed that it may prevent the viral entry from the endosome to prevent the endosomal fusion and release of the RNA, or it may protect the fusion of the viral supercapsid with the cellular membrane. So the chloroquine can prevent the release of the viral RNA from the endosome or from the surface of the cell. Okay? So then, some antivirals are under study that may prevent replication of the viral genome. There are several types of the drugs of this mechanism that are already developed, and of course, first the researchers group did. They start trying the antiviral drugs developed to prevent the RNA replication that were developed for other viruses. They start trying to inhibit the SARS-CoV-2 replication with these drugs. Not of them, of those studies, were equally successful, and a lot of them are under study. Then, when we discuss the therapy, so we will see what kind of drugs are more effective. Then by now we don't see or we don't have any pronounced strategies to block the viral packaging or the viral release. But once the viruses are released, of course, the strategy to neutralize the virus with antibodies looks very promising. Because truly, and you know this is a very old approach to treat infectious diseases. And this is a very old approach to treat viral diseases. So that's why plasma from reconvalescent patients is being used already for passive immunity, and some of the preparation used for this are on the third stage of clinical trials. It means large-scale trials in humans. So, but then again, there is the difficulties with the approval of this treatment in different countries. So then, and of course, the approach is done to manage management of the cytokine released within the immune activation of the immune cells that may prevent the excessive inflammation, my dears. So all those approaches have or aimed different have different aims, right? So, but the antiviral drugs aim at the inhibition of the or decreasing of viral load in the body and inhibition of the virus replication. So, few words about COVID, and I will just pronounce what I think you know. I hope you know already. So most people infected with COVID-19 virus will experience mild to moderate respiratory illness. And usually they recover without any uh, requiring special treatment. Of course, we have a very large group of people that have some factors predisposing to severe diseases. First, of course, this is the aged people, right? Then, some people, some children, we don't know why in some children it may go very severe yet. So, but of course, we have to consider those that have very severe underlying medical problems. And these are cardiovascular diseases, the type 1 diabetes, but also type 2 diabetes, then chronic respiratory illness, the cancer. Those underlying medical problems are more likely to develop, SARS-CoV-2 virus to develop very serious illness. So we will discuss right now. See, this is the picture where we can see the viral load dynamics and the duration and effectiveness. That is really very important issue. So that influence at when and how we can be infected. So after the initial exposure, patient typically develops symptoms within first 5-6 days. So this is the at the between -5, this is a type of time of entry of the virus into the body, and from -5 to 0. So for 5-6 days we have incubation period. See, 5-6 days when the symptoms are not seen. So then, since that time, the virus generate a very wide range of the types of the illnesses, from very mild, moderate illness, then severe illness with hospitalization that need hospitalization and accompanied with shortness of breath and acute respiratory distress syndrome. So this is designated as ARDS. And then, except for the severe illness, it may develop critical illness when the ACQ admission is needed, and that may result in the death. So, in patients with mild infections, initial host immune response usually is capable of controlling the infection. So, and we can see here that usually the antibody immune response will start to be developed within 7 days since the infection. 7 to 8 days within the infection. 5 days here and approximately 3 days here, right? So, in severe diseases, excessive immune response leads to organ damage. So, you can see here, we have the progression of antibodies, but then we may see the very severe disease in the presence of the antibodies. They won't really neutralize the virus completely. Then one more marker that is shown on the chart. This is the viral load. And you can see here, that is the viral load. The presence, counts of the viral genome in the blood. And you can see here that the peak of this, and yeah, what is important? So the virus multiply very quickly, and it will be seen and shed from the body within the pre-symptomatic phase and in early symptomatic stage. Or even if the infection is very mild, the virus shedding will be seen. So, and then one more thing, the virus shedding is not correlated directly with the severity of the illness. The viral load peaks in the first week of infection, and then declines thereafter gradually, while the antibodies response gradually increase. So, as soon as antibody response increase, so the virus will disappear from the blood. So then again, I would like to draw your attention at this. So in pre-symptomatic stage, and usually several days before the symptoms appear, and early during the symptomatic stage, this is the time when the person is most infectious. So since 6, 7 days of the disease, the patient stopped to be very highly infectious. So most probably the number of the viral genomes in the body or viral shed from the body are dramatically decreased. Okay, so now we'll start with pathogenesis, but then before we'll discuss the most probable ways of transmission. So what are the routes of transmission of SARS-CoV-2? Of course, you know, it will be transmitted by infected respiratory droplets. So, and it is potentiated during the prolonged stay in crowded, poorly ventilated indoor settings. My dears, I think more than 90% of cases are gotten in indoor settings. Then the next way is direct or indirect contact with the nasal, conjunctival, or oral mucosa. So then I would like to underline that social distancing and mask and the wearing of masks are really very important in decreasing over the possibility of virus transmission by these very important routes. Then one more thing. So the increased transmission occurs, and it had been shown where the large family collections, right? And when they eat together or drink together and communicate together very closely, my dears, that's a lot of restrictions is done for family gathering, because this is a very important way how the one can be infected. So I would draw your attention to the gastrointestinal tracts, because in the small and large intestinal tract, we have a lot of the cells of these tracts. So we have a lot of cells supplied with ACE2 receptors. So these tissues are very susceptible to infection. So we have information that it can be transmitted through ingestion. So then few words about the resistance or sensitivity of the virus. So it is known already that virus remains viable for many days on smooth surfaces, stainless steel, plastic and glass. And at lower temperature, so this occurs at lower temperature and humidity. And in the high humidity, the air droplets that are very important in the virus transmission may stay in air for a long while. While if we have the dry environment, then these droplets shrink very quickly, so and the virus can lose its infectivity. So then, regarding the sensitivity of the virus, it is readily inactivated by commonly used disinfectants, and that is true for all enveloped viruses because their envelope is very fragile. So, high potential value of surface cleaning and hand washing is shown because of the ability to inactivate the virus with the disinfectants. Okay, we go for pathogenesis of the COVID-19. And you can see here that S1 subunit on the spike protein binds with the AC2 receptor in the lungs, and these are mainly type 2 pneumocyte cells, pneumocytes cells, yeah. And it makes, so what is the pathogenesis of this exactly point? So in lungs. So the blocking of these AC2 receptors increase the production of angiotensin itself by the related enzyme, angiotensin converting enzyme 1. And the increased production of this angiotensin 2 potentially increase pulmonary vascular permeability, right? And may cause lung injury. So we may see pulmonary edema, dyspnea, acute respiratory disease, and acute lung injury at that moment. Then as a result of the antigen present, I think you've heard this picture. As a result of the SARS-CoV-2 activation of the antigen presented cells, so macrophages and dendritic cells, that lead to excessive release of the pro-inflammatory signals. What are the major inflammatory signals that are shown here, like in alveoli, how the antigen presenting cells, dendritic cells, and mainly macrophages are activated and release their cytokines. What are the cytokines? All interferons, alpha, to some extent, beta and gamma. The very important one is interleukin 1 beta. Then interleukins 6, 8, 12, 18, 33, tumor necrosis factor, and transforming growth factor beta produced by T regulatory cells. And also a lot of chemokines that are called CCR, CCX, and so on. They, by their release, we have the disbalance of the interleukin profile in the bodies that is now called cytokine storm. You see the tornado. And these inflammatory mediators may cause further damage of the epithelial cell lining and reach into the bloodstream and cause the damage to the other organs. And you can see that the damage can be done by this over-release of cytokine to the liver, and as a result the rising of the level of liver enzymes in the blood, then to kidney, and cause some effects in the blood. Then we will discuss it a bit. So, and that is a schematic representation of the immunopathogenesis. But then this is the immunopathogenesis, and you can see, and I have demonstrated it during the immunology lecture, if I'm not mistaken. But it also shown that the immunity will be developed. At the beginning, the immunity is doing very well, and the high level of interferons and all this help in containing the virus. But then later on within the disease, the dysregulation of the cellular and humoral immune response may lead to the tissue damage. So we take a look at the summary of the immunopathology. And you can see here the 4 groups of potential mechanisms. So, and the very important one, and I would like to underline this, the depletion and exhaustion of the lymphocytes. The poor prognosis of the disease have the direct correlation with the depletion of the lymphocytes. Then increase of neutrophil share in the blood. and they are very good in net formation, neutrophil extracellular traps formation, and release of harmless interleukins. Then cytokine storm, mediated by antigen-presenting cells at the beginning, and then T lymphocytes that later the disease may be really very damaging to the body. And also we mentioned that antibodies also may mediate antibody dependent enhancement of the tissue damage, antibody dependent cytotoxicity. So now a few words about the clinic of the disease. So my dears, there are so many clinical features and it is difficult to divide them somehow and to cover them all. Of course, you will be having the corresponding lectures in the therapy department, in infectious disease department, where more emphasizes will be done to the clinic. But then in the frame of the microbiology department, of course, you also need to know about some clinical features of the disease caused by SARS coronavirus. So we just look through it, okay? So, and the researchers, because of the studying of the lot of the cases of COVID, they divide the clinical features into common and less common. So common that have share more than 50, that can be seen in more than 15% of cases. That's what we discuss right now. But of course, we have a lot of non-common clinical features, and they take place in less than 50% or 15% of cases. So we take a look at the common. Fever is very common because it appears in more than 80% of cases. Cough is very common, in more than 60% of cases. Fatigue is seen in more than 35% of cases. Sputum production in more than 30% of cases. Shortness of breath occurs in more than 15% of cases. My dears, that what you see here is the result of particular studies. Of course, you can find in the internet somewhere. So a bit modified the list of clinical features, okay? So because that was the description of another study. So you should understand now that these figures are not really ideal. Of course, everything has been changing very quickly right now in our insights onto the virus and the infection caused by the virus. So the all thing that we see in the internet are the preliminary results because we live just 1 year with this disease. Do you remember that first the Chinese authorities claim the appearance of new human virus and the epidemic caused by this virus on the 31st of December 2011, right? Yes, 2011. So 1 year passed of our life together with COVID and SARS coronavirus. So those that's why some of the figures and the name of the interleukins and the sophisticated mechanisms of the cytokine storm and name of the cytokines. Of course, this been changing permanently right now. So I just give some, some basic cover of it. So then, the primary cause of mortality are respiratory failure. So we take a look at the causes of the disease, of the deaths. So why people die as a result of SARS coronavirus infection. So around 70%. This is a respiratory failure. Around a bit less than 30%, this is a sepsis or multi-organ failure. Around 14.5% is due to the cardiovascular system failure, and around 4% is because of the renal failure. Not so rare, but it also takes place. What some laboratory findings may be helpful in diagnosis of COVID? Of course, you know that first is computer tomography scan. And you can see here the abnormalities. And this type of abnormalities occurs in 86.2% of cases. Of course, this is understandable why computer tomography right now is considered to be one of the most effective laboratory methods. But then you have to understand that this what we see is not really highly specific. It can point at COVID, but it cannot be given as a final result of the diagnosis, right? Okay, so the most common pattern of abnormalities observed in the CT scan, this is called ground glass opacity, and with a very thick red line, I try to show this is, and can you see here, this is appearance that is called ground glass opacity and patchy bilateral shadowing of the lungs. Okay, so except for computer tomography, what other biochemical findings can be helpful? So these are lymphocytopenia, that is seen in 83% of individuals. Thrombocytopenia, so you decrease of the cell counts, 36%. Leukopenia in almost 34% of cases. So the very high level of C-reactive protein also is found, and we mentioned that liver damage may occur, and the result of it can be alanine transferase and aspartate aminotransferases liver enzymes levels will be elevated, and that can be seen by laboratory findings. Now we come up to the laboratory diagnosis of the disease. My dears, in many countries, we blame the laboratory diagnostic tests for uh, uh, false positive or false negative results. And this is understandable because there was not enough time to develop very specific and uh, uh, sensitive tests. So, but I would like to start with the least specific, but very rapid. You know, for any of the dangerous disease, usually the rapid tests are developed. So that let us within few hours or even 10 minutes, as it is pointed here, so to know whether it is the disease that we are looking for or not. So this is a comparatively new technique based on golden nanoparticles. Can we see nanoparticles? Golden nanoparticles. So, and this is performed after the sample collection from oral and nasopharynx, where we might have the coronavirus. Then it is combined in the liquid, in the buffer solution, with golden nanoparticles covered with the, we don't know what, because that is the know-how of every test. And then if the reaction is positive, is negative, nothing change in the color of the buffer solution. And if reaction is positive, we may perform naked eye detection of COVID-19. So the aggregated cold the nanoparticles will be aggregated, and they will change their color from yellow to I would say violet or so. So we, by naked eye, we may see the positive reaction. That's sort of test that can be formed in the airports. So that may give the result very quickly. So this is not really laboratory viral testing. These are express or rapid tests. They are always developed so to give false positive reaction but not false negative reaction. But my dears, nobody really knows how specific it is. Do you understand this? Because not so many testing had been performed. So quite often those tests can be blamed. For example, I read in Guardian that in the London airport, the rapid test missed 30% of SARS-CoV-2 positive individuals. Those that were shedding virus and had quite high viral load in the blood. Everything is understandable. A lot of people working on this to improve the tests. So, but by now, unfortunately, many of them may have insufficient specificity and sometimes sensitivity. Okay, then laboratory diagnosis. So, my dears, this is laboratory rapid testing is not laboratory diagnosis. This laboratory diagnosis. So, all tests can be classified into several groups. First, so-called viral testing, my dears. So a lot of students call it viral testing, but they think that we find live virus there. No, my dears. Viral testing is performed by the reverse transcriptase PCR test, and it used for qualitative detection of the nucleic acid for SARS-CoV-2. Not a live virus, but nucleic testing. So it is called viral testing, but we find really the viral nucleic acid. For these swabs are usually taken not from the nasal cavity, but the sample should be taken, the swabs, from the nasal, nasopharyngeal, and oropharyngeal. So swabs should be taken with one single, I mean, this swab, right? So then the sputum can be taken of lower respiratory tract aspirates or nasal washings. The positive test indicate the presence of SARS-CoV-2 RNA, and together with the clinical picture, these may support the diagnosis, but the negative test result do not preclude SARS-CoV-2 infection and shall be interpreted in light of the clinical picture and epidemiological information, my dears. That is the very sad. So the negative test doesn't mean that you 100% not infected with SARS-CoV-2. That is the truth for right now. Then the second approach that is used in diagnosis is serology, so-called. The test can assess prior exposure to the virus and cannot be used in the diagnosis of the current infection. Cross-reactivity with other human coronaviruses may occur. That is also not so optimistic because as we see now, coronaviruses are widely spread. So we may have human coronaviruses that won't cause any severe diseases. So they are almost harmless. But then they will give cross-reactivity in the tests. Then the serology test is particularly useful in some cases when, first, when the viral test is not available. Using the serological test together with the clinical picture could guide in decision making about the cause of the disease. Then, when the patients with late disease complications and their physicians need to make immediate decision about treatment, the viral test takes more time to get the result, and the serology may take, two hours for the result, 2-3 hours. So PCR tests takes more. Then, third, in some patients, in which virus shedding is reduced and making RT-PCR results falsely negative. So the serology test can detect IgM and IgG antibodies against SARS-CoV-2 in serum, plasma, or whole blood. So all these three types of specimens can be used. So rapid antigen testing. Uh, for this, we use monoclonal antibodies against SARS coronavirus nucleocapsid protein. And can you see it here? And protein. Why N protein? That's because this protein is abnormally expressed in the infected cells. The huge amount of N protein is released. So the monoclonal antibodies are specifically directed against nucleocapsid protein, and by using an ELISA, it is possible to detect SARS coronavirus 2. And the test has a reported sensitivity of 84%. Still, to compare with other infections, is not so high. But the specificity of the test is 98.5%. So it means it will not give false positive reactions. Sorry. No cross reaction with human and animal coronavirus in this assay were reported. But my dears, because the human monoclonal antibodies are used, there are no reports yet about applying this test to SARS coronavirus 2 diagnosis, I mean large-scale diagnosis. This is the antigen testing that is in process, so it is not available yet. So now, as soon as we diagnose the disease, we again take a look at the preparation that can be used for treatment. And of course, in immunology lecture, we have started with immunological treatment. And in lecture in virology, we start with antiviral drugs or so-called virus-based therapy. So with virus-based therapy, we try to inhibit the particular stages of the replicative cycle. And here I mention only those approaches that are under second, third phase of clinical trials, because it will take a whole lecture just to mention all types of drugs that are really now is under study. So that's why I will mention here that what is used really in the protocols for treatment. So, and among them we have protease inhibitors, and these are lopinavir developed for other RNA, other viruses, other viral proteases, and darunavir. And they inhibit the viral replication in the human cell. Then the viral fusion inhibitors. And the major one here is hydroxychloroquine. So the anti-malaria drug that, as we said, inhibits the lysosomal acidification and fusion of the viral RNA into the cytoplasm. And it prevents virus entry in vitro. So we don't really know whether it's, by the way, I've seen several studies where they showed no effectiveness of hydroxychloroquine. So there's a really very contradictive information about this. So, but it is incorporated into some clinical treatment protocols. Then the entry inhibitors, unfortunately, they are in progress. None of them are licensed, so we are not talking more about them. Then again, I would like to give you this again. You've seen it in the immunology lecture. But I would like to insist that immunological treatment is very important. So we will have here interferon-based immunotherapy, So with alpha, beta type 1 interferons and lambda type 3 interferon, and the function is direct, non-specific antiviral activity of these interferons. And then I would like to underline antibody-based treatment that we call one of the oldest historically approaches in treatment of infectious diseases. In the human monoclonal antibodies are used in a clinical trial that perform neutralization of SARS-CoV from the B cells of convalescent produced from B cells of convalescent patients. And they usually target the S1 subunit of the spike protein. And intravenous immunoglobulins that are taken uh, from the reconvalescent patients also and concentrated. But so they act according to the several mechanisms because the antibodies taken from reconvalescent patients may inhibit the Fc gamma receptors to decrease the antibody dependent cytotoxicity. Then there can be some antibodies neutralizing cytokines and antibodies suppressing the activation of lymphocytes, and also some substances that inhibit the function of the complement system, so antibodies against components of the complement system. So, and finally, we have the drugs for management of the cytokine storm. So, and here we mainly have the blockers of interleukin 1 beta, gamma interferon, interleukin 6, and basically corticosteroids that inhibit the expression of pro-inflammatory transcription factors in the nucleus of inflammatory cells. Okay, guys, I think with this we may finish. And this is the last lecture in your course of special virology. So there will be no other lectures delivered. So, my dears, do you hear me? This is the last one. So, with this, I would like to say goodbye to mostly students because I won't communicate them anymore because the exam this year will be given through Moodles and tests will be given. So, for the final exam, please, not CP, but final exam, and it is said now already that all students, irrespectively of how they studied, should give final exam. The final examination will be given by tests, first level tests with one single correct answer through Moodle. I think Dean's office will inform you, but from behalf of the microbiology department, I would like to tell you that you have a base for these tests because 60 to 70% of the tests will be taken from part 1 and part 2 department lecture books. Do you hear me? So please learn the tests from part 1. This is a general microbiology, and part 3 special microbiology. But of course 30 to 40% of tests may be similar but not exactly of the exact design with that given in your lecture books. Please, in your base, okay? Please, when you answer the tests, try to think. Try not to memorize the ABCD, but try to understand the question that you ask. That is my last suggestion. With this, please keep the social distancing, wear masks, stay healthy. Good luck on examination. Goodbye, my dears. Очень